Okay, so my name's Dagan Segal, and I'm a postdoc in the Danuser Lab. And I'm going to share an approach using xenograft to cancer cells in zebrafish to study cancer cell adaptation. And I've included here in my title slide as an example 3D renderings of cancer cells imaged in a zebrafish and classified by cell shape. And immediately you can appreciate there's a large diversity of cell shapes we see. And my long-term goal is to really study how cell shape in cancer can drive signaling and in that way drive cancer cell adaptation. And this idea that shape can inform disease state has been used for decades by pathologists, of course, in diagnosing and stratifying disease in cancer. And with the advancement of machine learning-based techniques, we now know that morphology can be an increasingly more quantitative correlate to cell state. Oh, you don't hear me? Oh, yeah, sorry. So a nice example is from the Wirtz lab here, where they see how certain morphotypes can really be predictive of transcriptional cell states or in vivo function of cells. But what's often missing from these state-of-the-art techniques is some sort of physiological context, right? We now know very well that even in culture conditions, it really matters if we're on glass or in stiff or soft collagen for both the morphology and the signaling state of the cell. And of course, in a tissue, things become much more complicated because we have interaction with neighboring tissues and chemical cues. And we also need to apply our analysis in 3D because cancer, unlike a lot of epithelial tissues, has really random orientation. So if we limit our analysis to just 2 dimensions and the orientations of the cell is completely random, we really miss a lot of information, right? And this is an example when we're looking at the same cell from different angles. If we just look at a 2D projection, we would get completely different information, right? We decided to address these problems that really zebrafish is an ideal model to study cancer cell adaptation because of its combined advantages of accessibility to high-resolution imaging, the throughput necessary to really capture heterogeneous data, and the complex microenvironments of a living organism. And here I've included 2 distinct microenvironments that I'm going to refer to throughout my talk. Namely the hindbrain ventricle, which is a fluid-filled compartment very rich in hormones, the paravital in space, which is this avascular region sandwiched between 2 membranes, and the caudal hematopoietic tissue, which is an area rich in vasculature that serves as a hematopoietic stem cell niche. So leveraging the advantages of this model, we developed a quantitative high-resolution imaging assay to study cancer cell morphology and classify cancer cell morphology in zebrafish xenografts. And I'm going to walk you through this framework a little bit, and this work has been published, so feel free to write down this reference and look it up for further detail or come talk to me. So what I do is I inject mixtures of fluorescently labeled cancer cells into different sites in the fish, and then using a high-resolution light sheet microscope, I image hundreds of these fish. And this microscope modality has the advantage of fast acquisition and the high axial resolution necessary for our quantitative analysis. And then because I have this mixture of fluorescently labeled cells, I can make out individual cell contours within a cluster of cells in the fish. And then I can extract those segment and extract those cell volumes and make some pretty simple 3D geometric feature measurements on those cells. And then finally, I can apply principal component analysis, and we see that our PCA actually captures quite well differences in morphology we do see by eye, as well as some we don't. For example, if we look at relative contribution of features, we see that PC1 is described by how protrusive the cell is, while PC2 can be described largely, but not entirely by volume. And then we can take this framework for various evaluation or visualization as needed. So we decided to apply this framework specifically to pediatric cancers, and more specifically to Ewing sarcoma, which is a a bone and soft tissue cancer. And Ewing sarcoma, like most pediatric cancers, is driven by a single fusion oncogene called EWS-FLI1, and very few additional somatic mutations. So despite the lack of accumulation of mutations, we really see differential patient outcome, right? 40% of the kids developed metastasis with very poor prognosis, which suggests, despite not accumulating mutations, which suggests that environmental drivers rather than accumulation of mutations probably drive these unique patient outcomes. So the experimental groups we chose to proceed with were specifically outlined here. So we're looking at these clusters of cells in the hindbrain of the zebrafish. So we have fibroblasts as a sort of non-transformed control, 2 Ewing sarcoma cell lines, and a matched pair of a Ewing sarcoma cell line, either with knockdown of the driving oncogene or a scrambled control. And for the rest of the talk, for clarity, I'm going to focus on this oncogene knockdown versus scrambled control pair. So when we look at 3D renderings of cells when we knock down the oncogene, already we can appreciate that the cells take on this more protrusive, more mesenchymal-like phenotype, and that's captured quite well in our PC analysis because we see a shift of these dots to the left in PC space, and we can visualize these in various ways. For example, if we use k-mean space clustering, we see that the knockdown of EF1 of the oncogene really leads to a shift to this more protrusive magenta cluster compared to the control cells. And this idea that oncogene levels can drive different states in Ewing sarcoma has been previously described based on cell culture studies, and that's led to a prominent model in the field where differences in oncogene levels can really drive a difference from a high proliferative state to a low oncogene expressing state that's more mesenchymal-like. So it's really nice to see this model recapitulated in our animal model now. But we really wanted, the goal of this was to study cell adaptation, so we really wanted to leverage the different microenvironments in our data set that I talked about earlier, namely the hindbrain ventricle, the paravitalin space, and the caudal hematopoietic tissue. So when we look within each environment, there's some heterogeneity of cell shape, right? And these are pseudo-colored according to the k-means plot I showed you earlier. But despite the heterogeneity, we really see nice tissue-specific specialization of cells. So this is the same PCA plot I showed you earlier, this time just displayed as a bag plot so we can recognize the individual experimental groups. So we see that the CHT cells tend to be more rounded and to the right in PC space, and the HBV and PVS are comparatively in the more protrusive cell state. And in particular, this result in the hindbrain ventricle was surprising to us at first because that is essentially a fluid-filled compartment, and the first intuition, you would think that cells within a fluid would be completely rounded, not protrusive. So we decided to examine this region further, and it was interesting. We actually saw an architecture, a tissue architecture that was unique to this environment, but not to the other environments. And that's the formation of these pseudo-rosette-like structures with cell bodies pointing outwards and protrusions pointing inwards towards a central point. And this was really exciting because it's reminiscent of a pathology in about 10% of Ewing sarcoma patients, which is termed pseudo-rosettes. So 10% of patients have these formation of rosette-like structures that pathologists define through this ring of nuclei. And we see these nuclear rings in our hindbrain ventricles, but not in the other environments. So it shows nice parallels between the human model and our zebrafish model, and also shows for the first time some evidence that this phenotype observed in patients is actually probably environmentally driven and not some sort of genetic predisposition. So now that we know that the oncogene affects cell state and that the different environments have some effect on the morphotype or cell state, we wanted to see if the effect of oncogene level could be modulated by the tissue microenvironment. So I already showed you that in our control shRNA, we really saw nice tissue-specific specialization of cells. But when we knock down the oncogene, we see this shift towards sort of a universally more protrusive cell state, and a loss of sensitivity to the environmental cues. Namely, the cells become sort of indiscriminately more protrusive, regardless of any environment they're in. So that led us to a model where we see, where we think the high oncogene-expressing cells are more stem-like, but also more able to respond to external cues because they're more plastic, while the low oncogene expressing are in a more differentiated state and less plastic. And we really think it's the ability of cells to sample all these different states, which enables their context-specific adaptation. And what's unique to the system also is that we can apply live imaging and analysis of dynamics of morphological changes over different time scales, right? We can look at time scale of minutes, how shapes changes within 10 minutes, but also how consistently a shape might change on the scale of hours, right? And there's actually differences at these different time scales. So when we look on the order of minutes, we really don't see a difference between our experimental groups. They sort of wiggle in some sort of morphological feature space. But when we look over several hours, we actually see a persistent shape change in the oncogene knockdown, but not in the control. So if we look at this framework more broadly, I'm excited about how it can enable single-cell profiles as a function of various factors of interest. I looked here at morphology, but we could look at other biosensors at a single-cell level, or we could characterize the tissue microenvironment in higher detail that interacts with these cells. Or what I'm most excited about and working on currently is similar to knockdown of the oncogene. We can look at different basal states that we might see in cell culture and see how they respond differently to different microenvironments. So, and I have a poster on some of the background in this project as well, and happy to discuss. So I'll end there. I want to thank my two advisors, Gaudens and also Jim Amatruda, who's now at USC, and especially my co-authors, Hania, who developed the computational framework for this, and Bo, who established the imaging framework that enabled this work. And I'm happy to take any questions.
Great imaging, and I have a question kind of going back to the oncogene knockdown. You picked sort of a key oncogenic driver, and I'm not as familiar with the pediatric mutation spectrum in this case. Is it sort of like a classic carcinoma? You have a sort of known oncogene driver and loss of a number of tumor suppressor genes as well, or is it very oncogene dependent cell line?
Yeah, so it's exactly the opposite, and that's a few pediatric cancers. That's what's unique to them, that they have a single driving oncogene and no accumulation of additional mutations, or very few and no correlation to those accumulation of mutations and disease prognosis. So it's really this single, very aggressive, it's a fusion oncogene, so essentially two transcription factors fused together, causing aberrant transcription, and that's enough to drive the disease. That's really, presence of this oncogene is what defines it as Ewing sarcoma.
Awesome, thank you very much.
Yeah, thanks, that was really nice. Just wondering too about once they're in that, say, the hindbrain niche there, and you showed the dynamics. Are you able to track? Are they moving around, or are they just kind of moving in place? And is it more bleb-like or effectin polymerization-driven dynamics that you're seeing? Or you can't tell.
Yeah, so I can image over 24 hours, and I was very surprised about how they don't move very much within those cell clusters. They really wiggle in place. They change their shape a lot, but I wasn't able to capture too much migration further away with those long-term imaging. And so specifically in the hindbrain, they tend to take on that specific elongated morphology towards a central point. In other regions, we have blebby cells. And what I've never been able to capture, there's a throughput limit with those 24-hour movies. The microscope's taken up for 24 hours on that one cell, so I don't have enough examples to make broad-reaching conclusions. But I've never been able to capture really a shift between a blebby cell to a mesenchymal cell, and it would be nice if I could capture that moment and understand it, but I've never actually seen that happen in vivo. I don't know what that means. All right, thanks.
Thank you.